Yeah, definitely. So that was kind of a different project from working on APOBEC, and in that we were trying to identify mechanisms of resistance to the HER2 ADC, Trastuzumab deruxtecan, or T-DXd. And we found an interesting observation that even though T-DXd has shown really high efficacy in HER2-low disease for like breast cancer, for other cancer types such as colon cancer, the effects have been kind of suppressed. So taking a closer look, we know that in colon cancers, EGFR, which is a HER2 dimerization partner, is highly overexpressed, and it does have an effect on the cycling dynamics of HER2. So we started with that information, and when we looked at it a bit further, we saw that overexpression of EGFR limited how much HER2 can get into the cells, and with this, it also limited how much ADC could get into the cells, and thus limiting the efficacy of the ADC in models. So what we found was a way to suppress EGFR using EGFR monoclonal antibodies that are already FDA approved, such as Cetuximab, and co-treatment of this Cetuximab with T-DXd actually brought in more T-DXd into the cells and rescued the efficacy of T-DXd. And this we've shown in preclinical models, both PDXs, cell lines, and now we're trying to design a clinical trial at MSK looking at the combination of EGFR monoclonal antibodies and T-DXd.